Hello everybody, good morning and good afternoon. My name is Mukesh Budania. I am PhD student at BITS Pilani. I am working under the supervision of Dr. Akash Chandrai. It is my great pleasure to present my recent research work on phytoremediation of airborne particulate matter in indoor environments. I can expect that everybody knows about the particulate matter air pollution. And if you see the global burden disease data of 2019, particulate matter air pollution is the second leading cause of premature deaths in India. Particles less than 10 micron in dia can get deep into the lungs that leads to diseases like respiratory disorder, digestive disorder, and even it may cause of cancer. As human beings typically spend up to 87% of their time in buildings, right now, because of pandemic, our most of the time spends in indoor. So my research is motivated by these certain discussed factors, and objective of my research is to quantify the role of indoor plants for the PM removal. To accomplish this objective, we selected three plant species based on their leaf shapes. First one is Christmas plant, that is needle-leaved plant, and second one is ficus plant, that is small leaved plant, and third one is croton plant, that is broad leaved plant. To conduct experiment with these species, we fabricated a plexiglass chamber with 210-liter air volume capacity, in which the temperature was maintained at 26 degrees centigrade, and the chamber was supplied air using mass flow controller, and we maintained the air exchange rate 0.5 hour inverse throughout the experiment. To maintain well mixed conditions, uh, we used three fans, two at vertical wall and one at the top wall. Temperature and relative humidity continuously monitored inside the chamber by using temperature and relative humidity monitor, and the size resolved particle concentration was measured by using a laser particle spectrometer. To measure the particle removal rate inside this environmental chamber, with and without the plant kept inside the chamber, we injected the insensitive particle from this PM exposure port and measured the particle removal rates inside the environmental chamber. We tested each plant species three times to quantify the repeatability of the experimental results. This graph shows the variation of normalized concentration of 0.25 micron size particles with time for the experiment conducted without the Christmas plant placed inside the environmental chamber. So from this graph, we can calculate the particle removal rate when Christmas plant not kept inside the chamber. Now in this slide, we can see what happens when Christmas plant kept inside the chamber. Red dotted uh, curve shows that when Christmas plant were kept inside the chamber, the particle concentration decayed much faster than that without the plant. This is because of particulate matter uptake by the plant surfaces. However, no such effect was observed when ficus plants were kept inside the chamber. As we can see in the graph, decay lines for experiment with the plant and without the plant overlapping, meaning that the particle uptake by the ficus plants was very small. In this slide, as we can see, the results for croton plant also similar to the ficus plant, or we can say that particulate matter uptake by the croton plant was also very less. From the decay data obtained for different particle sizes, we also estimated the size resolved deposition velocities, which is analogous to heat transfer coefficient. Here we can see that deposition velocity obtained for broad-leaved plant was near to zero. But deposition velocity is significantly higher for the Christmas plant as compared to the other plant species, because Christmas plant has needle-shaped leaves containing mucus oil on its surfaces, which creates favorable conditions for particle deposition. The deposition velocity were also found to be nearly constant with the particle size for the different plant species, because the particles generated by the incense stick had a narrow size range. As Christmas plant was most efficient to remove particulate matter, we estimated the average deposition velocity for Christmas plant, and using this we calculated the required number of plants for achieving desired percentage reduction in PM2.5 for the indoor environments. It can be seen that as the requirement of PM2.5 reduction increases, the number of plants also increases. For an example, for achieving 10% reduction in indoor PM2.5 concentration, the required number of Christmas plants are 44. But when requirement of PM reduction increases, for an example, if requirement is 50% PM reduction, then requirement of number of plants will be 400. As we are seeing at a constant air exchange rate, as the desired percentage reduction in PM2.5 concentration increases, the requirement of plants also increases. However, one more important thing can be noticed, that as requirement of plants also depend on air exchange rate. For an example, at an air exchange rate 0.5 our inverse, requirement of plants will be 44, and if we air exchange rate is 2.5, then the requirement of plants will be 150 for the same percentage reduction of PM2.5 concentration in a small residential room. So number 150 is infeasible for a small residential room. So it can be concluded that the Christmas plant was found more suitable than the ficus and croton plant. So for a small room, about 35 to 44 Christmas plants suitable for the reduction of 10% particulate matter level. But it also seen that indoor plants only feasible for the indoor settings with low air exchange rate. So these are the some important references here. Thank you.